Good morning. In this session, we will be focusing on how muscles contract, how they shorten, how actin and myosin play very important roles in the process of muscle contraction. As you know, in order for any muscle to contract, it has to receive a nerve impulse, and that with few exceptions, of course. We have seen the exception of the sinoatrial node, of the AV node, and all these modified structures in the heart that can generate electric impulses on their own because they have unstable membrane potential. But for all muscles, for all ordinary muscles, they should first receive a nerve impulse. And by nerve impulse, we mean a propagating electrical activity or a propagating action potential. So we will be discussing this in detail in the nervous system, inshallah. But when the nerve impulse reaches a muscle fiber along a motor nerve fiber or a motor neuron, this is followed by contraction of the muscle. But of course, there are certain conditions for the muscle to respond to a nerve impulse that we will be discussing later on. So, whenever a muscle receives sufficient nerve impulses or adequate nerve impulses, it will contract. But when the skeletal muscle or the cardiac muscle or the smooth muscle receives nerve impulses, every individual muscle fiber will receive a nerve branch. So as you see here, we have, for example, skeletal muscle fibers or myofibers. And if we call them, for example, 1, 2, 3, 4, you will be noticing that 1 receives a nerve ending, 2 receives a nerve ending, 3 receives a nerve ending, 4 receives a nerve ending. So when the muscle is innervated or when the muscle receives nerve fibers, each muscle cell or each myofiber will receive an individual nerve fiber. In addition to the nerve fibers, the muscle fibers will receive a blood supply, of course. They will receive arteries to supply them with oxygen and to supply them with the necessary glucose for glucose to be broken down and to release the energy and ATP molecules that are required for contraction. And this is the process of cellular respiration. And after cellular respiration is done, there are waste products like carbon dioxide, for example, that are released, and those waste products should be removed by means of veins or by means of the venous drainage of the muscle. Inside every muscle cell, we have contractile filaments or contractile filaments like actin and myosin. As you see here, actin is the thin filament or the thinner one, and myosin is the thick filament. So actin is thinner than myosin. And as you see, the actin filaments consist of, every actin filament consists of multiple bead-like structures. They look like beads. And they have specific sites here where the myosin heads can bind, as we will see later on. Myosin consists of parallel filaments, as you see in the diagram, and from those parallel filaments, there are protein molecules that arise from the myosin molecule, and that are called the myosin heads. So actin and myosin are both protein filaments that lie, that's why the title is actin and myosin filaments. And as you know, they lie inside the myofiber. They lie inside the sarcoplasm of the myofiber. And they are arranged in a parallel regular way, as we will see on the next slide. And they are responsible for the formation of dark bands and light bands through their particular regular arrangement. But not only are they responsible for dark and light banding in striated muscles, like the voluntary muscles and the cardiac muscles, but they are also responsible for contraction, for shortening of the muscle. So actin and myosin are those myofilaments. Actin and myosin are characterized by, there are common properties for both actin and myosin. First of all, myofilaments consist of actin and myosin, actin being the thinner one. The characteristic shape of actin is that it consists of bead-like molecules with specific sites on every bead for binding of the myosin head. The characteristic feature of myosin filaments are the myosin heads, but both lie inside the sarcoplasm of the muscle. And both are responsible for banding, for the dark and light banding of the striated muscles, meaning that in the smooth muscles, we still have actin and myosin, but they are not regularly arranged. Because if you want to have dark bands and light bands like this, meaning that if the muscle has to be a striated muscle, the arrangement of actin and myosin should be a regular arrangement. It should take a regular pattern. But if, like in the smooth muscles, actin and myosin do not have a particular regular arrangement, then they wouldn't be banded or they wouldn't be striated. The third common characteristic of those myofilaments is that they are responsible for muscle contraction. How? By shortening the muscle. And how do they shorten the muscle? We will be seeing that they shorten the muscle through sliding of actin filaments over myosin filaments. So let's check the electron microscopic structure of a myofibril, one of the myofibrils that are embedded in the sarcoplasm of a myofiber. So again, I emphasize that this is the electron microscopic structure of a myofibril, and that myofibril is embedded with many others, of course, with many other thousands of other myofibrils. They are embedded in the sarcoplasm of a myofiber. They are embedded in the sarcoplasm of a myofiber. As you can see here in this picture of the myofibril, that we have a dark band starting here and ending there, and that you have another light band. The dark band, which is this band, okay, is called the A band, whereas the light band is called the I band. So for every myofibril, we have a dark band that is called the A band that starts here and ends there. And then on both sides of the dark A band, we have a light I band. Okay, in the middle of the dark band, we have a zone that is called the H zone that lies in the middle of the dark band or of the A band, and that is less dark than the rest of the A band. If you focus more on that electron microscopic structure of the myofibril, you would find that, one second. Okay. If you focus more, you will find that this A band or that dark band consists of two types of filaments. The first type of filament is the thin filaments, these ones, and the ones alternating with them are thick filaments. So here you have the myosin filaments, and here you have the actin filaments. So it is clear that in the A band that starts here and ends there, we have both myosin and actin. And by definition, the A band is the band where, or the band or the part of the myofibril where you will have both myosin and actin filaments. What about the H zone in the middle of the A band? The H zone consists of myosin only. So it is dark because what gives the darkness is essentially myosin, because they are thick filaments, but it is less dark than the rest of the A band because it does not contain both myosin and actin, it contains only myosin. But if you look laterally to the right here and to the left there, you will find that in the I band, you have actin filaments only. If we enlarge this more and look at the molecular representation of the myofibril, this is how you can see the myosin filaments with their myosin heads under the electron microscope. And this is how you can see the actin filaments. And as you see, we have here this part starting here and ending there that corresponds to the dark band or the A band. And in the center here, starting here and ending there, we have the H zone that contains only myosin and no actin, whereas in the rest of the dark band to the right and to the left, we have actin, myosin, actin, myosin, actin, and myosin. So in most of the A band, in most of the dark band, we have myosin and actin, except in the H zone, the central part of the dark band where we have myosin only. What's present in all parts of the dark band is myosin. Whereas in the light bands, we have actin filaments only. As you can see here, we have actin filaments only, the blue filaments. Where are the actin filaments and the myosin filaments anchored? They are anchored to a complex protein structure present here to the right and to the left. That is called the Z disc or the Z line. The Z disc or the Z line is a specialized protein structure where to the right and to the left of the light band where the actin filaments are anchored. I'm highlighting the Z disc. And the Z discs are very important because they are the demarcation of what we call the sarcomere. So starting here and ending there, this area that is enclosed between two Z discs or Z discs or Z lines is called the sarcomere. And the sarcomere is very important because the sarcomere is the functional unit of a skeletal muscle, because every sarcomere alone can contract. Functional unit means, just like we said in the kidney, that in our study of the kidney, that the nephron is the functional unit of the kidney because every nephron alone can do the function of the whole kidney. Similarly, we can say that the sarcomere in a skeletal muscle is the functional unit of the skeletal muscle, or of the muscle in general, the striated muscle in general, whether skeletal or cardiac, because every sarcomere alone can contract. So the sarcomere starts here and ends there. And now we can demarcate the sarcomere here. It starts here and ends there. So if you want to know what a sarcomere consists of, so the sarcomere consists of an A band or a dark band in the central part, and then consists of half an I band to the right and half a left band, an I band to the left. What about the other half here? It belongs to a new sarcomere. So this is sarcomere number 1. This is sarcomere number 2. This is sarcomere number 3. This is only a part of sarcomere number 2 because here the dark band is not seen. Okay, the diagram is not complete. And there, this is only a part of the sarcomere because the dark band is not shown. So every functional unit or every sarcomere consists of a dark band in the center with two halves, two I band halves, one to the right and one to the left. Here we can see one sarcomere. So the green line here is the Z line. The green line there is another Z line or Z disc. The thick brown lines here are the myosin filaments, and the thin ones are the actin filaments. And this is a relaxed sarcomere, a relaxed sarcomere. This diagram represents the sliding filament theory, or what we call Huxley's theory. You needn't memorize the name. And this is a relaxed sarcomere, whereas this is a contracted sarcomere. So what happens actually is that the myosin heads anchor themselves to the actin filaments, and then they start striking them like this in order to, they start giving them strokes, hitting them in order to pull them towards the center. So it all starts by myosin heads that get activated, that anchor themselves to the specific sites on the beads on the actin filament, and they start striking them, giving them hits, giving them strokes in order to pull them towards the center. So at the end of the day, it is actin that slides over myosin. Myosin filaments remain fixed in their places. It's only their heads that attach themselves on both sides to the actin and start pulling them step by step towards the center. So actually it is the actin that slides over myosin, or the actin filaments slide over the myosin filaments until, as you can see, the sarcomere gets shortened. So a contracted sarcomere is a shortened sarcomere. And if all the sarcomeres are shortened at the same time in a long muscle, because the myofiber does not contain only one sarcomere, it contains hundreds of sarcomeres or even thousands, depending on the length of the myofiber. So if every functional unit and every sarcomere contracts and shortens because of sliding of actin over myosin, then the whole myofiber will shorten, and that's why the muscle shortens when it contracts. So the myosin filaments have oval heads, as we said, and the actin filaments look like a twisted strand of beads. And when a nerve impulse stimulates one muscle fiber to contract, every muscle fiber needs its own individual nerve impulse to contract. The heads of myosin will attach on specific points or beads on specific receptor sites between the beads of the actin filaments and pull them inwards in order to shorten the sarcomere. And then once they are done, once they pull the actin filaments towards the H zone, that becomes narrow, by the way, as you see in the previous diagram here. We had a very broad H zone. This one is the central H zone, and see how the H zone became narrower just because the actin filaments got closer to each other. So the sarcomere is shortened, and if there is synchronized shortening, meaning that all the sarcomeres shorten at the same time, so the whole muscle will shorten, and that's why muscle contraction is associated with shortening of the muscle, movement of the joint, and meanwhile thickening of the muscle, because when all the muscles all the sarcomeres get shortened and the muscle fibers get shortened, whereas the muscle has a tendon on both sides that gathers the muscle fibers together, then it is the central part of the muscle that will bulge. And as we said before several times, it is the insertion that is pulled towards the origin, meaning that when the biceps muscle contracts, this is the movable bone, and therefore it is the radius that is pulled towards the humerus or towards the upper arm, because the insertion is the tendon that is connected to the movable bone, whereas the origin is connected to the fixed bone. In order for all this to occur, as you know, the muscles require energy, just like any other cellular activity in the human body. For example, muscular contraction, active transport of substances against concentration gradient, and all this kind of stuff. We see that we need ATP molecules. The ATP molecule, as you know, ATP is, as we will see very soon, is adenosine triphosphate. And it needs to be hydrolyzed. It needs to be broken down by the addition of water in order to release energy. Why is the energy of ATP needed? The energy of ATP is needed essentially in order to allow those myosin heads that have been attached to actin to be detached from actin. And therefore, if you don't have ATP, then these heads will remain attached. So if this is a myosin head, and if this myosin head is attached to the actin filament, this is the attached head that will cause the stroke in order to pull the actin filaments towards the middle line, okay? But in order for that, so this is an attached head, and it has to stay attached in order to pull the actin filaments medially through the stroke or through the hit. But if it remains attached, if this head stays attached, then the muscle will stay contracted, and therefore you need ATP. You need ATP in order to allow that muscle, that head of myosin, to be detached. And if the head of myosin is detached, actin filaments return to their original position. And if they return to their original position, then the muscle will lengthen again and the muscle will get relaxed. So like all cellular activities, muscle contraction requires energy supplied by ATP. And this energy is essentially needed in order to detach the myosin heads from the actin filaments. But when the muscle contracts and relaxes, it does this several times, meaning that if you look at that, for example, if you look at that biceps muscle, and suppose that that biceps muscle needs to remain contracted for 30 seconds, don't think that the myofibers that have been stimulated will stay contracted for 30 seconds. No, during those 30 seconds, the individual myofibers contract and relax, but very rapidly, so that you cannot feel the contraction, you cannot feel the relaxation in between. So as long as you are giving nerve impulses to the biceps to stay contracted, then myosin heads will hit actin and cause shortening, and then relax, and then contract and relax. So myosin heads attach and detach, attach and detach. So because myosin heads must attach and detach a number of times during a single muscle contraction, and that statement is very important, a number of times during a single muscle contraction, then muscle cells must have a continuous supply of ATP. So what would happen if we don't have a continuous supply of ATP? Then myosin heads would remain attached to the actin filaments. The muscle would remain permanently contracted. Of course, this is extremely painful. And if your muscle stays permanently contracted, we call this tetanic contraction. So tetanic contraction is a continuous contraction of the muscle that results from ATP deficiency. Okay, of course, this is extremely painful, and this prevents you from doing a new action of the muscle. This is the structure of ATP. This is a nitrogenous, of course, it's all organic, meaning that they contain carbon and hydrogen. So ATP is an organic molecule. It consists of a nitrogenous base, and we call it a nitrogenous base because it contains the amino group, and therefore it contains nitrogen. And that nitrogenous, you don't have to study the formula, you just have to identify it. So adenine, and then it is connected to a pentose sugar called ribose. And then here we have 3 phosphate groups attached to adenine and to the ribose. Adenine alone is called adenine, but adenine plus ribose are called adenosine. That's why we call this molecule adenosine. And then because we have 3 phosphates connected, we call it adenosine triphosphate. The last bond here between the last phosphate group and the first 2 phosphate groups is a high energy bond. And because it is a high energy bond, then if we have an adenosine triphosphate molecule and we hydrolyze it, meaning that we break it down by the addition of water, the phosphate will be broken down, and we will end up by having, instead of having adenosine triphosphate, having 3 phosphate radicals, we will have adenosine diphosphate that has only 2 phosphate radicals, because the 3rd phosphate has been separated by hydrolysis, and a good amount of energy will be released. And now comes a very important question, which is, when your biceps contract, for example, if it is contracting for 1 minute, if you're lifting a heavy weight for 1 minute, the biceps has to remain contracted. but do all muscle fibers contract at the same time? In the biceps, no. They alternate in order not to get fatigued. Why? Because if you consider an individual muscle fibers, meaning that if you consider an individual myofiber, the individual myofiber obeys the all or none rule, meaning that it either contracts maximally at its maximum length, maximum strength, meaning that it shortens to the maximum, or does not contract at all. If you send electric impulses, if they are adequate, if their intensity is adequate, the myofiber will contract maximally. If they are not, if their intensity is not, then the myofiber will not contract at all. This is what we call the all or none rule, meaning that either you do the thing 100%, you do your task 100%, or you don't do it at all. So if this rule is true, if the all or none rule is true, then why are there differences in muscle contraction strength? Meaning that if you lift a pen, for example, you need low intensity contraction. But if you lift a heavy weight, you need your muscle to contract with more strength. So how come that the muscle can change its power of contraction or strength of contraction if every muscle fiber contracts maximally or does not contract at all? The answer is that the all or none rule applies to individual muscle fibers, but does not apply to the muscle as a whole. Because if we suppose, for example, that the biceps muscle consists of 1,000 myofibers, then if you want to lift a pen, then only 50 out of the 1,000 myofibers will contract maximally. But if you want to lift a heavy weight, a chair, for example, then 900 myofibers will contract. So for every myofiber, it will contract maximally or not contract at all. But what makes the difference in strength of muscle, the muscle as a whole, not the myofiber, power, is that not all the fibers contract at the same time. And this is important because it helps some muscle fibers to have some rest and to replenish their stores of ATP. So the all or none rule, muscle contraction is an all or none response. Individual muscle fibers either contract maximally or do not contract at all. So how then does the strength of muscle contraction differ from one action to another? The force of muscle contraction is determined by the number of muscle fibers that are stimulated. As more fibers are activated, the muscle strength will increase. And then the last thing we will be discussing is muscle tone and muscle cramps. When we contract a muscle, as we said before, it is partial contraction, meaning that not all muscle fibers are contracted at the same time. Rarely does the muscle use all its muscle fibers to contract. When it comes to anti-gravity muscles, meaning that if you stand upright like this, then there is, if this is your nose, this is the front, so you have a tendency to lean forward and fall because of gravity. So all the time, your back muscles have to contract partially in order to prevent you from falling forward. So when you're upright, the main burden is on the anti-gravity muscles that lie at the back, and they are continuously in a state of partial contraction. That's why when you stand up for long, you need to lie on your back in order to have some relaxation, because when you lie on your back muscles or your anti-gravity muscles no more need to contract. This continuous state of partial contraction, of contraction of a certain proportion or a certain percentage of myofibers, is what we call the muscle tone. And the muscles that have the greatest tone in your body are the anti-gravity muscles that lie at the back. Because these muscles are continuously contracting, even if partially, then they are they need a continuous supply of energy. They might use or they might use up all the available energy. If the muscle uses up all the available energy, like you do, for example, in a heavy match or in a difficult, stressful match, then what happens is a depletion of ATP. The muscles remain contracted for a while, and this is what we call the muscle cramp. And in order to prevent muscle cramp, those muscles like the end, of course, if you get a cramp in a match, you have to rest immediately in order to replenish your stores of ATP. But if in the back muscles, if you stand up for long and your back muscles contract maximally, they would use up all the available energy. That's why the muscle fibers in the back muscle alternate in order not to suffer from a cramp and in order not to use up all the available energy. Thank you so much, guys. And I'll be sending a worksheet on related to the stuff I've just explained. Thank you, and see you, inshallah, on Tuesday. Bye.